So this is like the second part, or let's say, yesterday we saw like an introduction to the concepts of super resolution. What I told you yesterday is like already not textbook knowledge, but more or less common knowledge on super resolution. And today I will show you some of the latest advances, and which are all aiming to surpass this 10 nanometer resolution, or to reach 1 nanometer or even below numbers of resolution, to try to reach true molecular resolution in biological cells eventually. So, but before we start, maybe we can discuss, or maybe we can have some minutes to have some questions about yesterday's talk, if you have any doubts. No? Okay. So we can skip all this. So I will show you now some research results from my group. So I'd like to acknowledge them. Here you see some snapshots of from a few years back. And here, more or less, the recent configuration of the lab. So we are a rather small group of physicists, chemists, and some biochemists working on these topics. But we are lucky to have a strong network of collaborators, both in Argentina and around the world. And the results I will show you today were made in collaboration with several people, with Tom Jobin and Stefan Hell in Göttingen, with Damián Refojo in Buenos Aires, Alfredo Cáceres, Nico Usain, Mariano Bisbal in Córdoba, Guillermo Acuña in the University of Fribourg in Switzerland, Philip Tinefeld in Munich, and Sabrina Simoncelli in London. Okay, so in our group, just to give you some snapshots, we don't not only do super resolution, we also do some nanophotonics using DNA self-assembly, and also we work on optical manipulation of colloidal nanoparticles. So I will skip this because we don't have too much time, but I will spend some time to explain a little bit of DNA origami, because we will use that in the talk. So DNA origami is a relatively new technology. It kind of started with this paper by Paul Rothemund in 2006. And the idea is to use DNA as a material here. You all probably know that DNA is a biomolecule that encodes genetic information. But here we are going to use just the property that DNA can form a double helix if it has the right sequence. And if it doesn't have the right sequence, DNA will not self-assembly to form the double helix. That's all. So now imagine that you have a single-stranded DNA, and you know all the sequence of that DNA strand. And now you let it hybridize with a small single-stranded DNA chain, an oligonucleotide, that is half complementary here and half complementary in another part of that long single-stranded DNA sequence. If you let that react, it will hybridize and form, it will fold the DNA locally, somehow like this. But now if you do this hundreds of times, with hundreds of small oligonucleotides with the right sequence, then you can fold this long single-stranded DNA chain into a predefined geometry. This is possible by design, by using the specificity of DNA. And you can do that in many different ways. I show you here this small rectangle, and here you see some examples of real life. So if you do this, you can prepare zillions of nano rectangles made of DNA by self-assembly. So they form automatically. It's wonderful. And on top of that, you can leave, for example, we call these small oligonucleotides, we call them staples, like the staples that put this thing together. So if we, for example, we take this staple here and we leave it, we make it a bit longer, then we can leave, for example, at this point, a single-stranded DNA chain that will be in a specific position of the origami, and it will be available for a subsequent binding of another DNA chain. For example, holding a fluorophore or a nanoparticle or something. So in that way, we can use these small rectangles as nano breadboards, as nano working tables to put fluorophore molecules, proteins, nanoparticles, and build stuff. Okay? So there are groups around the world that work on designing origami in very complex structures. Like you can see these nano machines, like here, all these three-dimensional structures, or combining different DNA origami to form larger structures, like in this case. So these are examples from other groups, just to show you that you can do many things with DNA origami these days. But in our case, we do very simple stuff. We just take, as I said, a very simple geometry and use it as a nano working table to put nanoparticles, proteins, particles, etc. And for example, here you see we use them to build a nano antenna for single fluorescent molecules. Here you see this gray thing with the shape of a T is a DNA origami designed to hold two gold nanorods parallel to each other and separated by a very small gap. Here you see in this transmission electron micrographs how they form. So these two rods separated by a small gap act as an optical antenna, just like these phase array antennas that were used for TV, you know, in the rooftops years back. This is a very simple design like that, just two elements. It's symmetric if the rods are the same, but if you put the fluorophore off center, for example, here, now you can have directional emission. So you can use this really to direct the emission of a fluorophore. Here you see a simulation, and we have experiments that show exactly this. So you can construct nano objects to manipulate light using DNA origami. We will use that for super resolution later, you will see. And also, super resolution. So DNA origami is useful for super resolution because we will prepare like calibration samples. But super resolution is also helping DNA nanotechnology, because if you construct complex DNA origami machines like these ones, and some of them are even dynamic, they move, you need some way to visualize that that thing is doing what you designed it for, right? You need to see how the DNA origami moves. And these are very small objects. So super resolution microscopy, especially with these very high resolutions that I will show today, is also helpful to check that your DNA origami or other nanomachines are working as expected. Okay. So let's get into the topic of today. So we saw yesterday super resolution microscopy is a real tool for discovery. We all know that. And we also saw that there is a limitation in the resolution that is not fundamental, but it's practical, and it's about tens of nanometers in the lateral dimension and 3 to 5 times worse in the axial direction. And that is the reason why, for example, we cannot see the supramolecular structure of microtubules, because the resolution is not enough. So we need to go beyond 10 nanometers to see that. Okay, so how to do it? The main problem, so the reason why super resolution methods cannot achieve any desired resolution is photobleaching, irreversible photochemistry. That's what limits the number of photons you can get from one fluorophore. And that's what in the end limits the localization precision or the resolution of your methods. So there are therefore two possible ways to solve this problem. You either get more photons somehow, or you either get more information from the few photons that you have. That's the two options. Well, we could add here, expand your sample, like you said, we saw yesterday. Of course, yeah. If you can make your sample larger, then there's no problem. But that's a separate way. Okay, so to get more photons, both avenues have been explored in recent years, and with success, actually. So to get more photons, for example, DNA paint is a very good strategy, but people have also developed exchangeable dyes, stabilizing buffers, self-healing dyes, and a bunch of strategies to obtain more photons from one position of your sample. And this works. The other way is where we have worked mostly recently, and it's about developing methods that can extract more information to determine the position of a sample or molecules with using few photons or some other strategy. And we managed to achieve this in 3 different ways. One is a STED FRET, so it's a combination of FRET imaging with STED. The other one is a very simple method to calibrate a TIRF microscope and obtain the axial position of a molecule very precisely. And the other one is what we call single molecule localization with sequential structure illumination, which is a family of methods that can deliver the position of a molecule with very high precision using few photons. So we will quickly go over these 3 ideas. And after, if you have questions, we can go deeper into the technical details. Okay, STED FRET. Yesterday we saw, for example, that if you use a STED or any other super resolution method, you can resolve this supramolecular structure in neurons, right? That are not visible in conventional imaging. This regular structure of actin, spectrin, and adenosine is supposed to have this structure. This is the working model. So you see, we believe that there are rings made of actin filaments stabilized by adenosine, and then spectrin tetramers make a spacer between actin rings, and those tetramers give the period of 190 nanometers. This is what we believe is the structure of this supramolecular assembly of proteins. Okay, so with STED we can image this periodicity. We can see that spectrin and actin are intercalated. We can also see that. But for example, we cannot confirm that actin and adenosine are in contact here, because that's a biomolecular contact. So we would like to, for example, apply FRET between actin and adenosine to confirm that they are interacting in this subdiffraction structure, in this very small structure. So you see here, this is the structure. If we do diffraction limited imaging, that's the resolution. We will not see this regular structure. If we apply STED, we can resolve it very nicely. But if we want to see biomolecular interactions in these regions, we need to apply something inside here. So the idea is to do FRET inside this subdiffraction volume. Okay? So to do that, people have tried different ways. People have tried approaches based on lifetime. It doesn't work so nicely because STED also modifies the lifetime, so it makes the analysis very complicated. So we went for an intensity-based FRET measurement that worked out, and I will show you how it's done. Basically, you need a 2-color STED microscope. That's all you need. And we first tried it using DNA origami, these things I just explained. So we built a DNA nanorod with a size of about 200 nanometers, and we placed, using DNA assembly, 2 FRET modules separated by 180 nanometers. That's more or less the same period of the natural structure. And we put 2 FRET modules. These modules have 2 donors and 1 acceptor. You will ask why not 1 and 1, and the reason why was that the photo bleaching of the donor was too high, so we just put 2 to be sure that we get at least 1 working all the time. Okay. And here you see measurements of a DNA origami sample. So in a confocal image, you see like diffraction limited spots. But if you do STED, then you see that each one of those spots is actually composed of 2, as you can see here. And you can detect donor and you can detect as acceptor instead, super resolve. So now, if you have a measurement of the donor intensity and the acceptor intensity, you can compute FRET. First, we observed it, by following in time. We let the molecules blink, and then we detected anti-correlated signals. When we do that in a diffraction limited measurement, the anti-correlated signals are very complex. Because we have several molecules blinking. But when we make a STED and we super resolve the 2 FRET modules, and we pick one, then in each one of those, we get clearly anti-correlated signals like in single molecule FRET. And here you see we had like 16 STED measurements on almost single molecules. So this is possible. Okay, so then we needed a formalism to compute FRET. from those intensity measurements. And for that, we use this formalism developed by Tom Jovin and Elizabeth Haref a few years back. And it's basically about computing this value F that is proportional to the energy transfer rate. So we are not computing a FRET efficiency. We are just getting a number that is proportional to the energy transfer rate. Okay? And to do that, you need to measure the intensity of the acceptor when you excite the donor, the intensity of the donor when you excite the donor, and the intensity of the acceptor when you excite the acceptor. These three measurements. In the pipeline, we include a confocal measurement of the acceptor before and after STED, just to account for photo bleaching. And that's basically it. Then the rest are calibration factors based on the spectral properties of the fluorophores. So in this way, you can obtain an image of this parameter F that is proportional to the energy transfer rate. Okay. But one disclaimer. This formalism that works very well was thought for ensemble measurements, where you have a clear concentration of the donor and a concentration of the acceptor, fixed. If you have that, this works perfectly. And that works well when you do diffraction limited FRET imaging, because you are averaging in your diffraction limited volume, you are averaging the signal of hundreds or thousands of molecules. But when you do STED in a sub-diffraction structure, in a protein assembly, you may have just a few molecules. And that means that when you measure in one spot and you move to the next one, the number of donors and the number of acceptors may be different statistically. And that will give you fluctuations of this FRET, of this F value, that do not correspond to real FRET, but just to statistical variations of donor and acceptor number of molecules, okay? So you need to account for that. And we did that preparing a control sample. That it was like we took microtubules and we labeled randomly alpha and beta tubulin with donor and acceptor. So, and then we made STED measurements. And in this way, we have an idea of what to expect of this F value when you have random distributions on a supramolecular structure of donor and acceptor. And this is what you see. These are the distributions of F that we obtained. And you see that when you do a confocal measurement, these two populations, the control and the sample, separate very well. But when you do STED FRET, two things happen. Basically, what happens is that the FRET events are determined more accurately. So you have volumes where you have very low FRET, and other volumes where you have very high FRET that in confocal imaging are averaged out. So here you have some very low FRET events and very high FRET events that are not visible here because you're not averaging so much. So nevertheless, you can put a threshold. Of course, it's arbitrary, but you can say, okay, whatever is above this, it's a positive FRET event. Whatever is below, I don't care. And like that, you can detect now FRET events in a sub-diffraction volume. And this is what you see here in this example. Here are neurons where we labeled adenosine and actin, and you see they are kind of on the same periodic structure here. And when you compute FRET, you see that you get high FRET signals with a periodicity that corresponds to the supramolecular structure. And if you do confocal FRET imaging, there's no way to discern any of this. So this is one way to obtain sub-10 nanometer information using FRET measurements and combined with STED. Okay, let's move to a totally different idea. So this is now we move towards direct measurements of molecular positions. So FRET is still a kind of indirect measurement inside that sub-diffraction volume. Now we go towards determining directly the position of molecules. And this method is basically about calibrating properly a TIRF microscope. It's very simple. So you know TIRF microscopy is about illuminating your sample with inclined excitation. And if your excitation angle is above the critical angle, then you get total internal reflection. if you have, let's say, a water environment sample and an oil immersion objective, right? In this case, the excitation light does not propagate into the sample. It penetrates just near the interface with an evanescent field that has this look. It's an exponential decay. So the intensity of the excitation field is stronger near the interface, and it decays exponentially inside the sample. Fine. But that's not the whole story. Molecules, fluorescent molecules, when they are close to an interface, they also change their emission pattern. So here you see calculations for a molecule that is perpendicular to a water glass interface, and here parallel to a water glass interface, as a function of the distance to the interface. So you see, for example, a molecule that is 250 nanometers away has this emission pattern. As the molecule gets closer to the interface, what we see is that the molecule emits more light into the higher refractive index medium. That happens for both orientations with a different spatial configuration, but in both cases, the molecules emit more light into the glass. That means into our objective, okay? So you can compute that. The green light is for a perpendicular molecule, the green curve is for a perpendicular molecule, the blue curve for a parallel one, and the red curve is the isotropic average. So you see that now we have two effects that make molecules brighter when they are close to the interface, okay? They are brighter because they are excited more efficiently, and they are brighter because they emit more light into your objective. If you combine the two effects, you can calibrate the intensity of a molecule as a function of the axial position. And this is what we did. Here the red curve is the perfect solution, the analytical solution, and in blue is an exponential fit. So you see that the dependency of the signal, the number of photons that a molecule will give as a function of the axial position, is nearly exponential. It's not the same exponential as the excitation field, okay? It's a different one, because we are taking into account also the emission, but it's an exponential that you can calibrate with three parameters. One is like the exponential decay, this alpha f, this df. Another one is this alpha f, it's a kind of background that every microscope has some background due to scattering of the excitation light. And the other one is the number of photons that a molecule would emit if it's exactly at the interface, okay? And that depends on the fluorophore and on the detection filters and detectors, etc. So df, alpha f, you can compute them based on your configuration. And N0, you have to measure it. But to measure it, it's very simple. You just throw on your glass some molecules, some labeled antibodies or whatever, and you compute the average intensity. So it's very easy to calibrate. And if you do that, you see the expected localization error is very, very small. Here you see what you get for astigmatic lens setup, as we described yesterday, using 7,000 photons. Here is another method called Donald, using the same number of photons. And here you see simpler, this method, using 5, 10, or 50,000 photons for different errors in this DF. Because this DF, you have to compute it, and you may fail a bit. But you see, even if you fail, the localization error is very, very small. Okay, so let's use it. So to use it, we need basically to determine this N0. So in this case, we threw a lot of antibodies on the sample, and we computed the average intensity. This was a DNA paint measurement, so we got a lot of photons, about 50,000 photons for each molecule. And then you perform a storm measurement or a DNA paint measurement, any single molecule localization measurement where you let molecules blink, and you know that you can determine the center of this, and you can tell the XY position. But on top of that, we take the intensity of that. And the intensity will give us the axial position. So basically, we take XY from a fit to this, and we compute the number of photons divided by the N0 that we determined here. It directly gives us the axial position, okay? The only care we have to take is that not to use the first and last frames of a blinking event, because in those frames, we are not sure that the molecule was emitting all the time. So in that case, we will be biased to a lower intensity, so higher fret, higher Z. That's it, the only care you have to take. So if you do that, now you can convert any storm data, of course, obtained in TIRF configuration, into a 3D storm data. And here you see microtubules, super resolved by storm in COS cells. And now we can obtain microtubule cross-section. So we can really see the cross-section of microtubules. And that is something that is not easy to do, I tell you. This is a very small structure. You need very, very sophisticated setups if you want to achieve this. And here you do it on a simple TIRF microscope. And here is an average microtubule of many of them. You can also detect microtubule bundles. For example, here in these neurons, that seem to be single microtubules. When you do cross sections, you see that there are like 4 or 5 of them bundled. You can see these kind of things. And also, remember that we wanted, for example, to achieve sub-10 nanometer resolution to try to detect the supramolecular structure of tubulin, for example, this periodic structure. Well, we tried. This is what you see here. These are plain cross sections of microtubules. We need to find a microtubule that is more or less aligned, right? That is not too curly. And then we can plot it and try to see if we see the structure. We don't see it, okay? We cannot see it. Why? Because we have the resolution. In this experiment, we have a resolution of about 8 nanometers, actually. But we don't see it because the labeling density is not high enough. And this is something that happens to all super resolution methods, right? We didn't discuss that yesterday, but one part of the problem is to obtain the resolution or the localization position. But the other part of the problem is to label your sample precisely. And if you want to see that really directly, we would need to label each single tubulin with an antibody or with a nanobody, and that's impossible. It's sterically impossible, electrostatically impossible. So the labeling density is not high enough. Nevertheless, we can compute, for example, distances to the first neighbors. And if we we take here a line, here a line, and we take in the XY plane or the XZ plane distances to the first neighbors, statistically we see that the probability peaks at about the period of about 8 nanometers. So you see that even though the labeling density is not high enough to visualize this directly, since we have the resolution, applying some very simple statistical analysis, we can infer that there is an underlying structure. And in this way, we can do kind of structural biology on a microscope, doing this kind of thing. Okay, so this is all basically about calibrating a microscope and a software. So if you're interested in applying it, all our software in the lab is open source, and you can download it here from this GitHub. And if you have any questions, we can, of course, you can discuss it. Okay, last part of the talk. And this is about yet another way of localizing molecules with high precision and using very few photons, few photons as we could. And we call this a single molecule localization with sequential structure illumination. So yesterday we saw that the usual way to localize a molecule is basically using a camera. You use a uniform illumination. If a molecule is fluorescent, you can get an image of it on a structure detector, like a CCD or an CMOS camera, and you get this kind of images, and you can pinpoint its position. Fine. The localization error of these measurements has this look. You see, it's basically in the numerator, you have the PSF, you have the diffraction limit, and you can make that error smaller and smaller the more photons you detect, okay? So, and you can get about 10 nanometers with a few thousand photons. That's the usual approach. A few years back, we tried to improve that, and we came up with this idea of probing the position of a molecule using a zero of light. So the idea was try not to, since we need to use very few photons, the idea was to use like a minimum of intensity to excite it as little as possible. And then we made a kind of triangulation. So here is we use this donut beam, like instead, but to excite the molecule here, not to suppress fluorescence, to excite it. And imagine this is the molecule. Then we excite it with this donut at 4 positions. And from those measurements, we could infer the molecular position, okay? We needed to use 4 exposures and not 3. You may think, well, it's a triangulation with 3, it should be enough. But the problem is that the donut is too symmetric. And then we needed a 4th exposure to kill some inconsistencies in the position determination. But if you do that, then the localization error still depends, of course, on the shot noise, no doubt. But now on the numerator, you have L. And L is the distance, the size of this excitation pattern. And if you use series of light, you can make this L as small as you want. Basically, you can zoom in and be more and more precise. Okay, I will explain this a bit more later. But this worked. You see, here using just 50 photons, we got an error of about 10 nanometers. And if we used a few thousand photons, then we already got to the 1 nanometer regime. This is just a simple experiment localizing a single molecule many times. And you see the histogram of localizations. And here you see the theory and the experimental precision. You see, we follow, of course, this inverse square root of n. But you see that now with a few thousand photons, we are already in the 1 to 2 nanometer regime. Okay. So this, at the moment, was I would say it was a breakthrough because we had this zooming in possibility. So you see, if you had, let's say, 1,000 photons, with an ideal camera, you would get to 5 nanometers. With a real camera, about 10 nanometers. And with min flux, we could get to 2 nanometers. And if that was not enough, we could shrink the excitation pattern and became more precise, reaching 1 nanometer or below. And we could use that for imaging and for tracking. Here for imaging, we had to test it. And then how to test this? Well, we use DNA origami again. So we prepare the DNA origami, and we put 6 fluorophores, or 8 fluorophores at distances of about 10 nanometers, and we image them. We let them blink, and we image one by one, and this is what we obtain. And you see, we can really image very small structures. The error bar here, the scale bar here is 10 nanometers. And for tracking, it's also beneficial because since you need very few photons to localize a molecule, you can track fast. Basically, that's the idea. In this case, we decided to be happy with 50 nanometers precision, a moderate precision. But to achieve 50 nanometer precision, we only needed 9 photons. So we collected just 9 fluorescence photons, and we got the position with 50 nanometer error. And that allowed us to obtain a diffusion coefficient at video rate. So this trace that you see here is not a position trace, it's a diffusion coefficient trace. So at 35 milliseconds. So each point here is computed from about 80 to 100 positions. And there you can see how these proteins were diffusing with some coefficient, then they got stuck, they diffused faster, then they got stuck again. And of course, we know where they stopped and where they are faster. So it gives really new opportunities for tracking. Okay. This was min flux. But then we continued studying this, and we had this, at the back of our head that this was something very simple. And when you start looking at the literature, you realize that people have been trying to localize molecules or particles for a long time. And people have tried many different things. For example, this one that was called full focus particle tracking is very similar to min flux, but they use a focused beam, a normal focused beam, like a Gaussian beam with a maximum. But it works. Or if you look at this is orbital tracking. Probably many of you know this because it was strongly developed in the group of Enrico. Orbital tracking uses a Gaussian beam around a circular path to localize the position of a molecule inside that orbit. This was proposed theoretically by Jörg Enderlein in 2000 and first shown experimentally by Enrico a few years later. And this has been used many times, many different configurations. Or if you look at this one, this was called single molecule confocal tracking. It was even published in Nature, and it's the same as orbital tracking. The same. Just instead of using a continuous readout, they used a discrete readout of with a few points. But it's the same idea, right? And then you say, how come these people didn't realize this, or we didn't realize that, or Minstead, published very recently. Minstead is again orbital tracking, but instead of using a Gaussian beam, they used a sub-diffraction, an effective sub-diffraction excitation using STED. So you excite a smaller volume using STED and then do orbital tracking, the same story. So, and there are many more. These are just a few examples to show you that over the years, people have tried hardly to localize a molecule or a particle as efficiently as possible, okay? And people have done it independently, trying their own ideas, their own setups, their own analysis routines. But what we came up after studying all this is that all these methods that look very differently can all actually be part of the same family. Of the same family that requires just three components. You need an excitation intensity that depends on position, okay? You cannot use uniform illumination. You need some information on the excitation field. This is IR. Then you need to shift that excitation field over a sequence of positions, RK, okay? You need to move it over space. And then you need to register, of course, the fluorescence signal of a molecule at each one of those positions of the field. These are the three elements that you need to make up many different methods, okay? IR, RI, and NI, the detecting fluorescence at each point. So with these three elements, the problem is to infer the position of a molecule in the vicinity of this trajectory. And we did that with maximum likelihood estimation, just to use the same estimation for all possibilities, because in the literature, people did many different ways. Okay, so now with these three ingredients, you can start building up methods. For example, the first thing you need to define is the sequence of positions that you will use in space. So you can use a raster scanning, right, for example, or you can do an orbital scanning, or you can do this weird min-flux scanning, or any other that you can come up with. It doesn't matter. As long as you don't use a rect line, which would be very stupid because it's too symmetric, you can use any shape of scanning, okay? The next thing is to define the excitation field. So the excitation field, if you're using focused light, there are basically two options. You use a maximum or you use a minimum. That's it. If you use two-dimensional excitation fields, you can have, there are more possibilities like lines, holes, any other thing. But for focused light, these are the two options. And now let's start combining them. So we can use a raster scanning and a focused beam, and we get this that is called confocal single molecule localization, which you would say is the easiest, right? But it was only recently shown because you need really fast scanners to capture the blinking of the molecules. Or you can do orbital tracking or Minstead with a maximum around a circular orbit, right? And this was done many times. Or you can do a min-flux, like use a donut, a minimum, and use this weird pattern, okay? All these methods were done, and they all work. But now you can come up with new methods. Why not doing orbital tracking with a minimum? It's possible. Why not? Or why not raster scanning a minimum? Right? You could also do that. So that's what we did in this work. We just tested all these methods under the same framework to make a fair comparison. And if you do that, this is like the optimum configuration, let's say, for each one of these methods. In this case, it's what you see here is the localization error using 500 photons. Okay? Here you see orbital tracking, this RASTMAX, min-flux, orbital tracking with a minimum, and raster scanning a minimum. The first thing you notice is that all methods using a minimum provide the lowest localization error. They are the most precise. But the second thing you should notice is that all these methods using a minimum, look at the scale here. They are all applied in a sub-diffraction area, okay? All methods using a minimum actually provide this zooming in possibility. You can apply them on smaller and smaller areas. But methods using a maximum, you cannot. That's the main difference. For example, for orbital tracking, there is an optimum size for the orbit. So if you make an orbit too small, it's like you're always playing with the maximum of your Gaussian beam, and you don't see differences if the molecule moves. Okay, so you're not very precise. So actually, the optimum orbit size is about the size of your Gaussian beam. And then if you play in the center, you have two steep intensity profiles that can tell you if the molecule moves. So that's why this was with a Gaussian beam of about 300 nanometers in width, and the optimum orbit is about 300 nanometers. Okay. If you want to do Rastex, you need to scan even a larger area. You need basically to scan not the width of your beam, but a complete PSF to really pinpoint the position properly, like we did with the camera. So that's why here is about 2 times the size of the orbital tracking area. Okay, but it works. But the key point is all methods using a minimum give you this opportunity of zooming in. And the reason why is because basically 2 zeros, you can bring them as close as possible. Or another way of seeing that is that a 0 is always a 0. You can go deeper and deeper into it, and it's always the same. And it always has information. If you approach a maximum, it gets flatter and flatter. The minimum is always the same. So here you see a parabolic minimum. If you dive into it and you zoom in and you expand that, it looks the same. And if you dive into it and you expand it, it looks the same. It's a scale invariant. So that's the key point. And then it doesn't matter how you probe that minimum. You can probe it with an orbit, or you can probe it with a min flux, or you can probe it with a raster scan. Okay? The main idea is to use a minimum. Well, in this paper, I try to explain these advantages in a didactic way in 1 dimension, to explain why you can bring closer and closer to minima, but of course this can be expanded to more dimensions. Okay, so from all these possibilities, this one was particularly attractive to us because it's a raster scanning thing, so you can apply it on any microscope that you could have, a confocal, a 2-photon. So we tried it. We took our scanning microscope, and you need to do 2 modifications. Like first, you need to modulate the phase of your beam to get a 0, and then you need to have a drift correction because these measurements take some time. And if you want to achieve 1 nanometer localization error, your sample should not move more than 1 nanometer in that time. But this all can be done. So here you see some single molecules. If you scan a single molecule with your donut, it looks like a donut, of course. And then what you have to do is to pick one and start scanning a small area. That's as simple as that. You start scanning a sub-diffraction area, in this case of 100 nanometers. And if you see, more and more photons, you see that minimum better and better, and you can pinpoint its position. And this is the localization error. In red is the Kramer-Rao bound, the theoretical limit. The gray shade is what we expect by combining the theoretical limit with our drift correction, which is not always perfect. So, and this is the experimental determination. So you see, we can reach 1 to 2 nanometer localization error with a few thousand photons. So then we tested it again to image using a DNA origami. So we prepared a DNA origami with 6 fluorophores. We put them on the microscope. The origami looked like this. We pick one, and we start scanning a small area, and we let molecules blink. So the experiment looks like that. We are scanning a 100 nanometer area on top of an origami, and we let molecules blink. And eventually one molecule blinks, and we see a minimum, and we determine its error by maximum likelihood. And like this, we collect positions. And after a while, we can render a histogram of positions that looks like this. And we really retrieve the geometry where the molecules were, again, with a scale of about 10 nanometers. So, and this is done now on any scanning microscope, okay? You don't need a very specialized microscope. Okay. How much time do I have? Or do I have time? Okay, very good. That's fine. Okay, so that's a summary of how much better you can resolve things with this kind of methods, okay? Now, I will spend the last minutes to tell you what I think is the real future of these techniques, because I don't think it's in imaging, honestly, because we are achieving really nanometric resolution. But these methods are very low throughput because we are looking at a very small area, right? It's like to zoom in inside the focus, and then we are very precise. But if you want to image something larger, the throughput is very low. And at the same time, DNA paint, these new fluorophores are providing so much photons, so many photons, that you can achieve also 3-5 nanometer localization precisions on a wide field microscope. And if you're labeling with anything, the labeling error is larger than this. So it doesn't make sense to go to sub-nanometer localization precision if your labeling has an error of 3 or 5 or 15 nanometers, right? But for tracking, this could be very, very interesting. And I showed you already some tracking experiments with the first configuration that we did that was using to do that. It was using electro-optic deflectors to do this very fast, but it was not so fast. It was 8 kilohertz. It worked, but we wanted to do it simpler and faster because this looks simple, but all these electro-optic devices are expensive. They need specialized electronics, etc. So a few years back, we came up with this configuration that is we call pulsed interleaved minflux. And the idea is to take a pulse laser, we split it in 4, we send those 4 beams through fibers of different length in order to introduce a delay of about a quarter of a period, and then we recombine the beams displaced in the sample. And of course, we modulate the phase of all of them to get a 0 intensity. And now we get, at the sample plane, we get this excitation pattern at megahertz, basically at the repetition rate of the laser. It's like a machine gun of measurements. It's doing, okay? And every now and then we get a photon. And since we use time-correlated single photon counting, every photon we detect, we can assign it to the correct donuts, okay? So in this way, we get like 4 times the lifetime decay, right? But if we integrate each of these curves, then we get the intensity produced by each of the donuts. And with that, we can compute the position. So in this way, we get position and lifetime at the same time, and we can measure very, very fast. And the setup looks complicated, but it's just mirrors and fibers. We don't need electronics or anything to control that. And here I show you some examples, what you can do with this. So first thing you can do, of course, is to multiplex by lifetime. So here you see DNA origami with 2 fluorophores that are spectrally equivalent, identical, but they have different lifetime. So now we can fit this with a 2 exponential, and we can determine the intensity of each one, and now we can localize the individual fluorophores with the same color, just by lifetime multiplex. Okay, that's very simple. Now we can use lifetime or of the speed of tracking to monitor dynamic DNA origami. I told you that you can design a DNA origami that also make functions. A very simple one is this kind of structure, like schematically shown here. It's a DNA origami with a long DNA arm that can bind in one position or another one, transiently, okay? And those 2 positions are separated, in this case, by 12 nanometers. This origami was designed to have an average binding time of 2 seconds, this one of about half a second. And here you see, you can follow in time that with the fluorophore at the tip of that arm, we can follow it how the dynamics of this. And we can clearly see the slow dynamics, the fast dynamics, and the 2 positions separated by 12 nanometers. And this can also be discerned by lifetime in this case. So we can really follow nanometric motion and multiplex by lifetime. And then we applied this to more rather complex structures, like we did these 2 arms that I showed you before. They were on separate origamis. But then we made an origami with these 2 arms are on the same spot in a sub-diffraction volume. And they are doing their own dynamics. And we can detect them. By the different lifetime, we can detect each one of them, okay, on the same spot. This one is very interesting and very nice, I think. Here, this is a work done with Philip Tinefeld, by the way. The group of Tinefeld is expert on building this kind of DNA origami structures, and so they're really wonderful systems. So you see, here they build a DNA structure with a flexible DNA bridge with 2 epitopes for an antibody. So when the antibody binds, this DNA bridge that is flexible will reflect the position of the epitopes with two fluorophores. So now by measuring these two fluorophores, we can measure distances between epitopes without disturbing the epitopes. Because one of the things is, yeah, I can label them here, but then I don't know if they are working or not. Here we can just indirectly measure the distance between two binding sites of a biomolecule. And this is another one that is nice, that here we have a lipid membrane with biotin lipids and streptavidin, and then we let DNA origami, we let it diffuse like a boat over there. And we have two fluorophores. So since we have two fluorophores and we can detect them simultaneously on a nanometric position, we can not only determine the position, but also the orientation. So we can see how this boat is diffusing and rotating on that membrane. Okay, and one last experiment, which I think is also very nice. This is a way of using the position and lifetime information that we have to detect the position of a dark absorber. Okay, so there's so many ways of detecting fluorescent molecules or bright particles, but how to detect a dark absorber? So one way to do it is, again, with in this case we use a DNA origami, but it could be with random adsorption, but in this case it's more controlled, just to show the principle. So we have a molecule that can bind at different positions near the absorber. And depending on the distance to the absorber, it will undergo more or less energy transfer. Okay? So the lifetime will change. So every time it binds, so we determine the positions by main flux, and we get the lifetime of each position. And in this case, we get, like, you know, for each position, we get like a circle of where to expect that absorber. And then we can, where the circles cross, we can determine the position of the absorber very, very precisely. You see here, with less than 1 nanometer error. By performing these measurements, we can locate like absorption centers. Okay. With that, I finish, and we can discuss whatever you like about this method. So I showed you this method called Simpler to calibrate a TIRF microscope and achieve sub-10 nanometer resolution in 3 dimensions on any single molecule localization measurement. The combination of STED and FRET that gives us information about biomolecular interactions inside subdiffraction volumes. And this family of methods of single molecule localization using structured sequential illumination. That I showed you, there are many methods, but all methods using a zero of light provide the highest resolution. And here are some thoughts about possible applications in the future. I think pulse mean flux is fast and accurate, and it's actually the fastest and more accurate method that is only limited by the count rate of the emitter. So if you use a single molecule, in fact, you don't get much better than other methods because the count rate is not high enough. But if you follow a particle or a vesicle with a lot of fluorophores, you can reach nanometric precision and microsecond time resolution very easy. And that is one of the projects we are aiming now in our lab. And then Rassmann and mean flux for imaging, as I said, I think this high photon efficiency of these methods is kind of losing significance because other ways to deliver a lot of photons became very, very successful. So it's like you have a lot of money, you don't care about spending, right? But it may be interesting to use it to apply super resolution with bad fluorophores. So, you know, over the time, many fluorophores, actually most of the fluorophores, have been discarded for super resolution because they were not good enough, especially fluorescent proteins. So I think a niche of application of this could be to use Rassmann or mean flux to achieve decent super resolution, say 10, 20, 30 nanometer resolution using bad fluorophores, or particularly fluorescent proteins. This could be interesting. And another interesting application would be to combine the two, because now we have two methods that provide nanometric precision to localize fluorescent species, but they both work physically using different phenomena, because camera-based detection is based on emission, and mean flux and Rassmann are based on absorption. So now you have two methods that probe different phenomena. So you can combine them to track energy flows, because you could probe where energy is absorbed and when energy is released, for example, in 2D materials, and to probe exciton diffusion in some nanostructures. Okay, with that I finish, and I'm happy to take any questions you may have. Thank you very much.